Hello and welcome back to Quick and Dirty Med School. Today we'll be going over histology. Let's start with the cytosol and the cytoskeleton. Of course, you should know that the cytosol is this kind of fluid matrix which bathes the organelles and helps maintain cellular shape and plasticity. As you can see here, it's actually quite crowded. It's not what most textbook pictures show, which makes it appear like the cytosol is empty and there's one or two things in it. But no, it's actually it's quite crowded. As you can see here, here's the cytoskeleton, and here are the ribosomes, and the cytoskeleton plays a huge part in maintaining a concentration gradient from intra and extracellular fluids, and it plays a big part in like cell signaling and allowing those molecules to come in and then interact with whatever's in here. Main compositions is water, oxygen, carbon dioxide, electrolytes, and proteins. And this is where glycolysis takes place. Remember that. Glycolysis takes place in the cytosol. And it's composed of the cytoskeleton gives it structure, which is microfilaments, microtubules, and intermediate filaments. As you can kind of see here, here's the intermediates. Here are the microfilaments. So microfilaments are made out of two kinds of actin. Globular actin, which polymerizes with fibrillar actin, and that plays a role in contraction, anchoring, and positioning proteins onto the cell membrane. Microtubules are made out of alpha and beta tubulin subunits. They're much more robust than microfilaments. They're stiffer. They play a bigger role in structure, and they also play a large role in the structure of the centrioles, cilia, and flagella. Colchicine, vinblastine, and paclitaxel are medications that interfere with the polymerization of tubulin. Intermediate filaments are the most stable, and here's a few examples of them. Vimentin are class 3 intermediate filaments. Those are found in the mesenchyme. Glial fibrillar acidic protein, those are found in astrocytes, and astrocytes are found in the brain, and these are cells that nourish and maintain the neurons. Lamins, those make up the lamina, and that's an important distinction. Lamins are the proteins. Lamina are the layers. Keratins or cytokeratins are found in all epithelial cells. So the presence of a specific type of intermediate filament in tumors can often reveal the cellular origin of the tumors. Like I listed above, they are quite specific. All right, the nucleus, of course, that contains our genetic information. This is where replication takes place. This is where ribosomal assembly also takes place. And cells can be enucleate, means no non-nucleated, like erythrocytes. Mononucleate, which means only one nucleus. Multinucleate, which is multiple nuclei. And binucleate. The locations can be basal, which means towards the bottom of the cell, central, in the very center of the cell, or eccentric, which is kind of like the cell is polarized, so the nucleus will be on one end and the rest of the organelles will be on the other end. Generally, the differences and variability of these like location and nuclei structure is in leukocytes. So the nucleus has its own version of the cytoplasm, which is called the karyoplasm. And like the cytoplasm, it supports the nucleus, creates that kind of electrochemical gradient, assists in the plasticity. In muscle cells, they have something called the sarcoplasm, which contains calcium ions, which are released whenever this muscle cell contracts. So perinuclear on the outside are the outer nuclear membrane, and that is continuous with the rough ER. And the inner nuclear membrane is attached to the nuclear lamina inside here, and those two are attached via the nuclear pores. The nuclear lamina is made of the lamins, proteins, and class 5 intermediate filaments. The nuclear pores are they contain nucleoporins, which is a protein, and the nucleoporins regulate diffusion because the nuclear envelope is also semi-permeable. Euchromatin is, as you can see here, electron-lucent. It is the uncondensed it's the uncondensed chromosome, and the heterochromatin are the condensed chromosome. All right, so histones kind of determine whether something's going to be euchromatin or heterochromatin. One nucleosome has an octet of these histones, and DNA winds around the histones. So as you can see here, it forms a structure like beads on a string. And then when this DNA is condensed into like heterochromatin, as you can see here, they're very tightly packed. So whenever it's time for like a replication and transcription, the transcription or replication signal will come in once euchromatin is a little bit unwound, because like here there's a little bit of a gap. There's a space that allows the signal molecules to bind and to start the process of replication or transcription. As you can see here, heterochromatin is up here, it's condensed. Euchromatin here is less condensed, and this picture didn't show, but the histone 1 is like all down the center, and it keeps them like it keeps it bound tightly together until the kinetochores are expressed, and then these two are able to separate. So the nucleolus is where ribosomal RNA is synthesized and the ribosomal subunits are assembled. So in the pars fibrosa, after things have been like transcribed, the newly synthesized, newly transcribed rRNA comes out and like assembles here in the pars fibrosa. And then down here in the pars granulosa, that's where the ribosomal RNA is linked to the proteins and the ribonucleoproteins, and they start accumulating there. The nucleolus itself is quite acidic and stains strongly with basic dyes such as toluene blue, methylene blue, and hematoxylin. So ribosomes, of course, assemble the proteins, and they are electron dense. They are found either in the cytoplasm or in the rough endoplasmic reticulum. Polysomes are they're aggregates of ribosomes, and they are attached to a single mRNA, and it permits the synthesis of like multiple copies of the same proteins. They synthesize like intracellular structural proteins, cytosolic proteins, transport proteins, and they're intensely granulated. So, and they're acidic, so they're basophilic, and as you can see here, like they are like homogeneous in appearance. Like it's like a very solid blob. The rough ER is only seen in the electron microscope, and the smooth ER is larger, so it can be seen with the light microscope, but is much better seen with electron microscope. Secondary lysosomes digest metabolic waste, cellular waste, whatever, and it is heterogeneous in appearance. Residual bodies are vesicles holding indigestible materials such as like pigments, and they are secreted by exocytosis, and they become like lipofuscin granules, as you can see here. Unfortunately, lipofuscin granules accumulate in long-lived cells, and they reflect aging. So they'll accumulate in neurons and skeletal muscle, and they appear yellow-brown in the light microscopes, and they're quite electron dense. Lysosomes are uniformular granular. They're electron dense. They're acidic. They are better seen though with UV fluorescence, as you can see here. This is it is marked with the red fluorescence. They are acidic, of course, because they usually contain peroxide to degrade various wastes. So Pompei's disease is an acid maltase deficiency, and it causes lysosomal glycogen to accumulate, especially in the heart and the muscles, and it causes severe muscle wasting. And the only treatment is enzyme replacement treatment. Lysosome defects could also cause lysosomal storage disease, and that's like just the toxic buildup in our cells. Tay-Sachs is quite a terrible disease, and it is a deficiency in hexoaminidase A, and that causes a failure for sphingolipids to degrade. So those don't degrade, and it causes accumulation, and it leads to severe neuronal degradation. Unfortunately, there is no true treatment of this disease. You can only really treat the symptoms. Peroxisomes are, they contain peroxide, like hydrogen peroxide, and they help with the degradation. And the difference between a lysosome and a peroxide is like basically where they are created. So a lysosome is made in the Golgi apparatus, and it buds off, and the enzyme in it are made in the cytosol, and the enzymes are the peroxisomes, and the peroxisomes are self-replicating, and like I stated before, it is made in the cytosol, and a peroxisomal targeting signal on the lysosome brings them inside. So peroxisomes, they lack nucleic acid, and any defects cause some severe demyelinating diseases. Adrenoleukodystrophy, it's a problem in transport. There is a disruption from the long chain fatty acid transportation. Mitochondria, as everybody knows, is the powerhouse of the cell, but it also has another role in degradation. So it delivers reactive O2 species to the phagosomes, and reactive O2 species are just oxidants. In electron microscope, you can see them a lot better. They are round, or they're rod-shaped. In the TEM, you can actually see the double membrane. You can sort of see it with light microscopes. This is not the Eosins dye, but but it can be stained with eosin. This just shows up a little bit better. But as you can see here, they're just tiny, 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 like little flecks. Mitochondria are the size of bacteria, so it's not very, very useful to see them under the light microscope. So back to the structure of mitochondria, the porins are proteins that are on the outside membrane, and those assist in the electrochemical gradient and plays a role in like the sodium potassium pump. And the cristae are like the inner foldings, and that plays a role in electron transportation. All right, the Golgi apparatus, you just know that it processes and packs, right? So it processes macromolecules by like glycosylation and phosphorylation, and they also process the proteins by activating them via proteolysis. And they pack them in vesicles and send them off to whatever structure in the cell that needs it, or they even like send them extracellularly. The Golgi apparatus is like, you know, flattened pancakes, as you know, middle school book states, but those flattened pancakes or stacks are called cisterna, and they are, you know, they're organized in cis, trans, meso, and endo sites. More importantly, the cis face is the nucleus, and the trans face is away from the nucleus. They stain poorly with normal light microscope. As you can see here, it's like this like very faint non-stained area right outside of right here the nucleus, but not particularly distinct. For the rest of the cell, cilia are 9 to 2 dynein and microtubule organization. Cilia has the 9 to 2 organization, while centrioles only have 9. Important distinction between the centrioles and centrosomes is that the centrioles is the one unit, right? It's the one subunit. A centrosome is, if you've seen my other lecture, it's 2 centrioles in a kind of like L-shaped pattern. And the chromosomes are, so chromosomes, pardon me, the centrosomes are like in place and they're active during mitosis or meiosis. Cilia and flagella aid in, you know, cellular movement. The difference is that cilia are much smaller and they move in a beating pattern, so the cells can kind of like inchworm their way around. Flagella are much longer and they move in a corkscrew motion. Last but not least, inclusions are non-living components made from the metabolic products. So they can be like free-flowing glucose, glycogen, like whatever metabolic products there are in the cell. Okay, that's it. Thank you for your attention and good luck studying.